Good morning everyone, respected professors and fellow students. Welcome to today's session of the summer internship program. As a master student from Jawaharlal University, it is my absolute privilege to welcome our esteemed speaker, Professor Rana Singh. Professor Singh is currently serving as the Vice Chancellor of Gautam Buddha University and is also a professor at the School of Life Sciences JNU. To truly understand the scale of Professor Singh's scientific impact, one only needs to look at his remarkable research output. His peer-reviewed work has accumulated over 17,700 citations. His work spans critical and complex areas of biology, including cancer stem cells, the use of advanced spheroids and organoids, DNA repair, cell signaling, and even microgravity radiation biology. Crucially, his research heavily focuses on cancer chemoprevention using non-toxic natural phytochemicals. His contributions to biomedical science have been recognized globally through numerous prestigious honors. Most recently, he was awarded the Government of India's International Fellowship for Senior Biomedical Scientist to Johns Hopkins University. This follows previous international recognition, including the ICMR International Fellowship to UC Riverside. These milestones are built upon a strong foundation of earlier career accolades from the American Association for Cancer Research and the US Department of Defense. We are incredibly fortunate to have a scientist of this caliber here today to demystify the complex world of cancer biology for us. Please join me in giving a very warm welcome to Professor Rana Singh.
Today we will try to understand about cancer. All of you might have different curiosities or questions related to cancer. So maybe first, if you can start with some questions, it will be better. I want this class to be interactive. So unless you ask question, I won't be like comfortable in telling that what I know or what we should discuss or what we should be doing. So just begin with the small questions. Any question which you feel that I should be knowing in the area of cancer biology. Anyone can ask a question. You can start.
So how people become
So question number one. How benign tumor become malignant?
So why till now we are not able to treat cancer?
So second question, still we are not able to manage the cancer or treat the cancer.
Why CAR T-cell can't use for the detection and the treatment for solid cancers?
Why?
CAR T-cells.
CAR T-cells. So CAR T-cells are being used currently for the
blood-related cancer.
cancers and for the treatment also.
So why solid cancer? Why we not prefer it for solid cancer?
Solid cancer. Why it is not being preferred for the solid cancer?
How M2 macrophages get transformed into M1 macrophages in the CAR macrophages.
So M2 to M1 conversion or vice versa. How it is there in the tumor microenvironment. So any other question?
So we have products like this, like protein target degradation. So इनके बाद भी हमारा like cancer managed to be, we have protein targeted. So हमारे healthy cell affect ना हो, तो उसके बाद भी managed to be.
So the question is related to differential sensitivity. Normal cell versus cancer cell. And still we have targets, but we are not able to control the tumor. So whatever questions you have asked so far, if you look at the publications, many labs are still working on this. Although we will have some answers related to that, but still people are working. So this tells that still we don't have complete understanding of whatever questions you have asked. I am sure that you have seen literature and you will find the papers which are being published in these areas whatever you have asked. Like your question was benign to malignant conversion. So how a benign tumor can get converted into a malignant tumor? So first we need to know what is benign tumor and what is malignant tumor. Once you know the differences, then you try to connect that if there is a protein or any gene which is the underlying cause of making a cell malignant, what is its status in the benign gene? Suppose you take any gene and the gene is functionally normal, but only it is overexpressed. There is no mutation. So you can take any proto-oncogen. So unless this proto-oncogen is getting converted into an oncogen, likely the cell will be only growing, but it is not going to be growing. This proto-oncogen at any time it can get regulated in a normal way, the way it should be in a normal sense. So that is how sometimes we see these kind of molecular changes, unless they happen, cells are not going to be converted into the malignant cells. So you will find a lot of details. So your question was related to the treatment. Why still we are not able to treat the cancer? So in this respect, you see there is a difference between communicable diseases and non-communicable diseases. So in communicable disease, mostly it is through the pathogens which are getting into the system and they are causing some disturbances, and from where we are seeing the symptoms of the disease. In cancer cells, mostly you don't see outside pathogens. It is your own cell, and somehow the cell is not under control. So once the cell is not under the control, and you are trying to manage it, to control it in normal. So if I just give you one example of one cancer cell, suppose you take colorectal cancer. So can you guess that when it is a stage 3 or stage 4 cancer, how many genes might have mutated in that cancer cell? Any guess? Just guess any number. How many genes you expect to get mutated or they are not functioning properly in the malignant cells?
Multiple genes.
I'm asking number. It is multiple. Any number you can guess? Just guess. I'm not asking exact number. You should tell some number, then I will tell where you are and where you understand how you feel about malignant tumor and malignant cancer. Any number? Don't tell any number. See, you are not getting my question. When I am saying guess, I am not asking, so try to understand what I am asking.
600.
So they are very close. And 400 to 600 genes are mutated in one malignant cancer cells. So suppose 400 to 600 genes are not under control. Can you imagine a drug which can fix 400 genes? Any drug, can you imagine, which can fix? There may be tumor suppressor genes, not working. There may be proto-oncogenes, there may be DNA repair genes. There are so many genes which are required for the cell to function normally. So this is the problem in cancer treatment. Once the cells are malignant, we are not able to handle them, as we don't have any drug which can specifically target all those genes to fix them. So this is main problem.
But sir, through surgery, we are still not able to completely cure the cancer.
See, surgery is the only way to have a sure shot treatment of cancer. But surgery will be successful only when the cancer is not metastasis. When you talk about malignant, it means it is all over in the body. Surgery is not going to work. So only when it is early detection, it is primary tumor placed in situ in the organ, then it is possible that if you surgically remove, then you can say the person is cancer free, and only when until it is not moved anywhere in the body. So that is the only way a person can get free from cancer. Otherwise, whether you have chemotherapy, radiotherapy, immunotherapy, biotherapy, all 10 plus therapies, no therapy is sure shot. Use any therapy, even if one cell is left, again it is going to recur and there will be formation of second. So other than surgery, there is no cure which can guarantee you 100% that a person has become cancer. It depends upon the system in the body, our body response to different kinds of therapies. Whether it is 90% successful or 100% successful. So this is the problem with therapy. Your question was related to differential sensitivity. So why most of these drugs are not working, or even if they are working, sometimes you might have heard that cancer patient died not because of the cancer, but because of the side effect of the treatment. The reason is that whatever drug therapy we have, they are targeting those proteins or genes which are also there in the normal cell. All of you might know this. So there is no selectivity. If there is no selectivity, then normal cells are dying, then side effect is going to happen. If the cells are dying in the critical organs, then you will see severe health threat. And that is why still we don't have a magic drug which you can say that will only kill the cancer cell and not the normal cell. And the reason is that all the genes are there in the normal cell as well as cancer cell. The only difference is in cancer cells you might be having the mutated ones. So once a drug which is very selective, very specific, which is going to target the mutated one, and again whether it is protein or gene, again that is going to make difference. If you are targeting gene, then likely you can have the outcome which is better than targeting protein. So if you are targeting protein, gene is still there, it will keep producing those oncoproteins. So it will be of no use. So these are some of the limitations why still we don't have very selective drug for normal cell versus cancer cell.
So, but for that selectivity, we use targeted drug delivery and adapt to kind of things.
Yeah, nowadays hundreds of labs are working on this aspect. How to have a selective drug which can target only cancer cells. But still we don't have any drug in the market. So still people are working how to make a selective drug. And again, as I said in the beginning, one drug is not going to work because you have more than 400 genes every day. So that is the second limitation why it is not going to work. What is your question?
Why CAR T cell is not effective in
CAR T cell. Okay. So CAR T cell, your son was also amount to M22. Okay, so once we go into the field of immunology, see these cancer cells, all you know that they are expressing different kinds of proteins on their surface, and some proteins they are supposed to make them abnormal or differentiated from the normal cells, and they are supposed to get recognized by the immune cells as an abnormal cell, and they should be removed from the body. But this is not happening. it is not happening. What could be the reason? The reason could be that the protein which is supposed to be there and recognized, somehow it is getting masked. Masking is happening. And cancer cells are having those mechanisms to avoid the recognition by the immune cells. So CAR T cells, they are engineered and they are from your own body, as you know. And they are engineered to just look into this problem and see how inherently they can reach and they can identify such cells and they can target these. So actually now this is in the clinical use. Earlier it was in the experiment. But in some cases now it is being used. Even in India there is one lab, ACTREC, with the help of IIT Mumbai, and they are generating CAR T cells and trying to have the treatment of the patients in the hospital. But still we need to know how much success rate is there. And only this is because of the difference where we have several types of markers on the cell. This is again going to come into. So these challenges are still there. And M1 to M2 is related to how the cells are getting polarized in terms of the demands of the needs in the body. So again, when it is a pro-inflammatory environment, when the tumor is going to grow, then you will see conversion. And pro-inflammatory environment is always required for the growth and progression of the tumor. So most of the tumor, they create their environment on their own. They will try to secrete some of those proteins or some of those ligands where you see these conversions are happening in different immune cells, where this one is also there. So it depends upon what kind of environment, what kind of drugs you are using to reconvert them and try to control. But again, the same problem will come. How and at what stage that cancer has reached. In the early stages, it might work to control up to maybe some extent. And that is what you see. In some cases, people respond better and their survival is more. In some cases, it don't.
Sir, but how the body comes to know that like M1 macrophages are the pro-inflammatory ones and the M2 are tumor suppressive ones. So they are meant for regulation. So how the body judges that the amount of M1 should be more or less?
It is all the signaling molecules, that is what I said. This conversion is dependent on what kind of signals are coming, either to M1 or either to M2. So tumors are smart enough to send those signals, make them pro-inflammatory. And that is how they create their environment. So signaling you might have read, there are several different types of signals which will be working in the favor of tumors once they are there. Or they are there. And it will be determined by the kind of cytokines they are producing. So pro-inflammatory cytokines, they will be there in tumor cells. So when you read the signaling pathways, then you will see at very minute level how a particular signaling is getting activated and how other one is getting inhibited. That is how you see the difference between one type to another type. It's a very careful regulation by the combination of the ligands which are being secreted either by tumor or by any other cells. So any other question?
So, is there any biomarker to diagnose cancer in early stages?
There are several biomarkers nowadays for different kinds of cancer, but still you might have heard in the hospitals they will test the blood or they will take the biopsy and they will go for molecular diagnosis. So again they are looking at different kinds of proteins or the genes or their mutation. So the biomarkers are there. your question is that how to detect it early. So why detection early is very difficult. I'll just give you one example. See, most of the tumors, they don't reveal or cause any symptom, which is very different from any other disease where they are growing. For example, let's take liver cancer. So I came to know about one case. One person was there in a village and he was having some stomach upset, digestion problem. So what people do, they will first change their diet, light diet, curd and all those things. But still it didn't improve. Then what you take? Diazine or those kind of medicines to just have a better digestion. Again, it didn't work, and like that it went on for years. It is intermittent. You take the drug, medicine, it will be okay. Again, after a few weeks, it will same thing, same problem. Then that person went to the clinic after a few years. That I am having this stomach problem from last few years, it's not being controlled. Then doctor suggested that you should do ultrasound. Then we got ultrasound done. So ultrasound, you might be knowing the principle how it works. It will work based on the density of the tissue. So if there is any dense tissue anywhere, you will see different kind of color on the image. So in ultrasound, doctor suggested that sometimes in one of the liver lobe, if there is any fibrosis or tumor, you can see clearly that it is showing different kind of image. That is what the doctor found. It looks like there is some problem in the liver. Then it suggested that you should go for the CT scan. So in that process, it took several years. A person from the village, and just what is the problem? Stomach upset, nothing else. So after several years, when CT scan was done, it was found that his liver was having almost 10 centimeter, 10 by 8 centimeter of tumor in the liver. So you can imagine how big the tumor was. So what was the symptoms? Stomach upset. If there is a brain tumor, what will be the symptoms? Headache, nausea, vomiting, migraine type of symptoms. So in any organs, there are several diseases which are of normal type. Whatever symptoms are there, you will find same symptoms even if the tumor is growing in that organ. So that is why it is very difficult to diagnose it early. Because suppose a stomach upset was there, if doctor in the beginning, if the doctor has told that person that you have cancer, what will happen? Without proper diagnosis. Or even for the headache, if somebody is going to the doctor, so unless proper tests are being done, nobody can predict. And usually now there is a tendency of avoidance. Even if we put a health camp over here for cancer diagnosis, if I ask you all 30 people to go and give your samples for diagnosis, I am sure that only few of you will be going there. You are very sure that I don't have anything. And the same situation is there in all ages of population. One is ignorance and other one is fear. Fear is that यदि कुछ निकल गया तो फिर क्या होगा? That is the fear. So people avoid. So unless we have a policy that we should get diagnosed at regular interval. If I tell you the policies in the Western world, in the USA, there is a policy that people should go, the male, and they should go for prostate cancer screening. And if it is female, then cervical cancer and breast cancer instead. So that is kind of mandatory. After 40 years of age, it is kind of mandatory. And why it works? Because in those countries, you have medical insurance that covers the cost for the screening. Currently, in our country, we don't have this coverage from the insurance. So unless it comes in the policy, insurance people are not going to cover that. They will cover once they will get admitted into the hospital, not for the screening of diagnosis. So that difference is there. So such situations are making the management of cancer very difficult. And let me just tell you about, I ask you a very simple question: can cancer be eradicated? What will your answer? We have a slogan that polio is eradicated. Similarly, if I ask, can cancer be eradicated? Why? So we have one answer, right?
The cause of the polio was only the polio virus. But the cause of cancers are so many mutagens, so many fluids and pollutants are
So any other idea which you can tell that why cancer cannot be eradicated other than what you told? Let me give you my hypothesis why it cannot be eradicated. See, all the living beings or even living cells, they have tendency to survive. This is inherent property for all living beings or all living cells. Tendency to survive. So when we talk about the cells, how they are going to survive, they will proliferate, they will make clones of their own. And they know that even in some adversity, if our existence is being challenged, at least there should be some cells to survive. Some can sacrifice, but some should survive. So that is how cells are proliferating. So it is designed in a way, if you look at the age, generally we have average age of 72 currently. And after 40 years, usually aging starts. If you look at the incidence of cancer, after 40 years it peaks like this, it's exponential increase. In 40 years it will be less, after 40 years it will go like this. So there is a correlation of aging versus incidence of cancer. What it tells that when our body starts aging, some cells have tendency to survive. Cell does not want to age. So in that process, cell is trying to acquire such mechanisms through which it will survive forever. What could be those mechanisms? To have mutations in those genes which can make them immortal. They are not going to die. So that could be the process why, as long as we have humans or any animals on this earth, cancer cannot be eradicated. What it means? That it is a kind of natural process, I would say. It is a natural process of the cells to resist itself from the aging. In that process, it is acquiring such mutations or changes which is making them transform from normal to cancer cells. Although they will also die with the person or with that animal wherever they are. But that is the process. Just to resist the aging, this could be one hypothesis. And if this is true, then it cannot be eradicated no matter what you do. As long as humans are there on the earth, cancer will also remain. Cannot be eradicated. There could be other hypotheses as well. But this is one what we see mostly when you look at the literature and the experimental outcomes. So any other question?
I think there is any technique we can develop to especially direct the cancer cells. So because the relapse is occurring most of the time because of the research of cancer stem cells that left behind after the therapy. If we detect them at the earliest, that if they are still there, we can target them.
So detection early before the treatment or after the treatment?
After treatment, some of the cancer cells, if they get remain, then they cause the reoccurrence of relapse.
So after treatment, always patients are on wait and watch and follow. If there is any treatment or surgery, doctors will ask them to come after 3 months, after 6 months, depending upon how it progresses. So that follow-up is there, also tests are there. Biopsies are taken, blood is taken. So tests are there to find out whether cancer is coming back or not, the relapse is there. So nowadays, technologies are advanced. They can figure it out, the cancer is coming back. But for those cancers which are coming back, it is very difficult to cure or control. Because that is going to be more aggressive. It will not respond to those earlier therapies. Because cancer cell has survived all those earlier therapies and acquired the resistance against those therapies. Even radiotherapy. So it is called a radioresistance, which is against the radiation. And there will be chemoresistance, which is against the chemotherapy. So resistance is being developed against all types of therapy. That is how these cancer cells are split. And again, this kills that survival. For their survival, they can acquire any such mechanisms. Any other queries? Okay, so let's go through some slides. I have few slides. This is about the biodiversity of the campus. You might have noticed so far. And this is where I am working currently. It's Gautam Buddha University. This is in Greater Noida, it's a state government university. And here we have 160 courses, 80 schools, 8,000 students. And several other facilities are there. Campus is very beautiful. Now let's look at this very simple data over here. X-axis and Y-axis. Y-axis you have age increasing from 4 to 85. So left side data is year 2000 and then so what difference you see in 20 years? This 20 years of difference. So this data is pyramid shape. Most of our population was younger in 2000. Now this younger population is moving. So it is here currently around our average age is 29 years in India. What will happen if you go for another 20 years? In 2040. This is going to move here. So you will find most of our people are going to be in 60s, 70s. So a lot of aging population will be there after 20 years. And In the beginning, we have already discussed that all the chronic diseases, they are going to be heavily burdening the country. The chronic disease, you have cancer, diabetes, cardiovascular, neurological diseases. All these diseases are going to be associated with the age, and we are going to have a huge burden. So we need to think early, plan early, how we are going to manage the burden of the disease. So this I just move. This is just prediction that cancer incidence and cancer death tells that still it is increasing. Means still we don't have enough strategy to manage the disease. So here we have A, B, C. A for apple, B for book, and C is for red pencil. If I ask you how it relates to the cancer, any idea? What is the ABC of cancer? So I will give you one example related to what is ABC of cancer. You know, you have car. In car you have accelerator, brake, clutch. अभी तो automatic हो गया है. So ABC is there in the car. Cells what we have? Tumor suppressor genes. This is analogous to the stop sign. And you have proton pulses. That is very much analogous to the green signal. Suppose you are driving the car with the speed of maybe 80, 90, and if any of these are not being functioned properly, then what will happen? Car will met with an accident. So there is a similar situation. You have tumor suppressor genes and proton genes. If there is some problem over here or here, then likely it is going to cause cancer. So accelerator and brake, you know, you have brake, accelerator, and clutch you can just assume as a DNA repair genes. They are very important. Any mutations are happening, DNA repair genes are not working, then these changes will happen. So those are the important ones. Okay, let's talk about a little different thing over here. This is related to concepts in Ayurveda. As along with the cancer, our lab also works in the field of Ayurveda biology. I just wanted to convey the concept. Just start from the right hand side. If you look into the ancient literature, Ayurvedic literature, you will find mention of these words, avyakt and vyakt. Avyakt is very much similar to subatomic level, and vyakt is atomic level. So you can assume here electron, proton, neutron. Here you have atom, and these words are already there in our text and described. This is what you study in the physics. Next you come, then you will find these words Tanmatra and Mahabhuta. This is equivalent to molecules and macromolecules. That is what is studied in chemistry. You come next, then you have Prakriti and Tridosha. This is what you study in the biology. Hetu, Linga and Aushadhi. Hetu means etiological factor, diagnosis and the treatment. So these concepts are already there. This is very much close to what we study in the modern science, physico-chemical, physiological and psychological attributes. Currently now some advanced techniques are there. That is how we are trying to understand using computational systems. So what I want to convey here that if you look at our ancient literature, these things were known, however the language is different. So unless we read them, try to understand, plan some experiments, validate them, we'll never know that what people were doing at that time. Because health management is there from the beginning. Not the only what we know from last 100 years, whatever developments we are seeing in terms of modern medicine, it's just 100 years of development. So all these concepts were there, they are very much equivalent to the modern medicine system. So just look at one ancient text, Charak Samhita. There you will find the mention of Granthi and Arjuda. If someone had read this, they might know it. So Granthi is used to be called for the benign tumor and Arjuda is for the neoplastic tumor. Similarly, there is a mention of progression of cancer, and that is also related to the imbalance in doshas or the involvement of inflammatory conditions. So these things are already known long back. This is again a very simple slide just to see how normal cell division is happening. And once we have a mutation in one cell, what is going to be its fate? Either cell will die or it will proliferate. So suppose cell is coming under proliferation and then it is dividing and forming the malignant tumor. So if I ask you a very simple question in this lower part, tell me about your one significant observation. What is happening in this case when it is getting multiplied into the form of tumor? One or two observations, critical observations. What is changing from here to here?
So at some stages, regardless of the mutations get accumulated more and more in the progressive stages.
Okay, yeah, that is good. So you are telling that this red.
As the generation passes on, the mutations get more and more accumulated.
So here 1, 2, 3, 4. The red ones.
Yes, sir.
What is other observation?
Apoptosis is not occurring.
Apoptosis is not occurring, or apoptosis is occurring.
In cancer cell it is not.
Any other observation?
Apoptosis is not occurring.
Even after the mutation is getting accumulated, the organisms are not working in apoptosis.
Any other observation? Very simple one.
Color and shape of the cell.
Yes. Another simple one. What is changing when you go from first mutation to second mutation to third mutation to fourth mutation? Tell me. Number. So actually, number is decreasing. I just wanted to hear this. This number is decreasing. So what is the relevance when the number is decreasing? There are several changes. This is very important. First mutation, you have 4 cells, just for example. And when you have second mutation, out of 4 only 3 is surviving. Third mutation, only 2 cells are surviving. Fourth, only 1 is surviving. And at this time, it is getting converted into malignant cells. So there was a hypothesis that at least 4 mutations are needed to convert a normal cell into a cancer cell. That's the old hypothesis. But what is more important over here is that at each and every mutation, the cell is facing a challenge for its survival. Because if there is a change in the cell, it is going to be abnormal ones. And we have immune surveillance mechanism in our body, along with other surveillance mechanism, to identify abnormal cells and remove them from the body. So this tells that from here to here, 75% cells are surviving. If it is becoming more abnormal, then only 50% cells are surviving. Here, 25% cells are surviving. So there is always a challenge in our body. If there is anything abnormal, it is going to be challenged. Same thing is happening to our cancer cells. So unless they pass all the challenges, they will not survive and they will not form the tumor. So getting cancer or having cancer, again it is not very easy for these cells. That is what is depicted in this picture. So succeed, sustain and survive. And this is the fundamental principle of any living system. Succeed, sustain and survive. Now answer this question. 1 plus 1 is equal to? Any other answer?
11.
Any other answer? 2 and 11. Now answer this one. Can you fill the blanks? So how many numbers you can fill there? What are the possibilities? So here 1 plus 1 is equal to 2. So you have just one possibility to get one answer. To get the same result, if I put 2 here and I am asking fill this blank, both the places. So here there are infinite possibilities. You also put the numbers in minus sign, there are infinite possibilities to get the same answer. So the idea is just to make you understand how biological system works. It is very different from the mathematics. Biology is very different from the mathematics. Very rarely you will find additive effects. Very rarely. If you combine two treatments, you are getting X effect from one, another one also X, you will never get 2X. But it will be lower or it will be higher. So that is what you have, either synergistic or less than that. And if they are working opposite way, then antagonistic. So you might have done your experiments in the lab, and you would have seen, I guess, when you do the treatments, when you combine two agents, very rarely you will get just double effect or just additive. Very rarely. You will find some differences. So biological systems are very complex. So we have earlier discussed that what are the main causes of cancer. We have chemical, physical and biological. All the cancer causing agents can be classified in 3 major categories. Polyaromatic hydrocarbons, nitrosamines. All of you know about chemical carcinogens, how they get activated and bind to the DNA, make adducts and do mutations and transform normal cell into the cancer cells. Physical is radiation. So what is the first evidence of radiation that it can cause cancer? There are historical examples.
Radium. And then Marie Curie discovered radium.
Madam Curie. Any other example? Major ones. Remember about atomic bomb explosion? Hiroshima and Nagasaki. 1946 or 45. So usually nuclear bombs, they are exploded about 800 meter to 1 kilometer from the Earth's surface, so that it will spread to the larger area. So in Hiroshima and Nagasaki, those people who directly exposed to the explosion, they died immediately. But those who survived on the periphery, it was found that the people were having incidence of cancer 3 to 5 times higher than those people who were not exposed to radiation. Another incident you might have heard about, Chernobyl reactor accident. It was in USSR that time. That incident happened. There also same observation was there. Those people who got exposed, they had 3 times more incidence of cancer. And the third historical example is related to non-nuclear proliferation treaty, NPT. Have you heard of them? Earlier, when USSR and the USA, they were having always Cold War between these two, because they had nuclear warheads very high in number. So that treaty was signed to reduce the nuclear warheads. It was NPT, non-nuclear proliferation treaty. No one is going to make more nuclear warheads. So in that process, there is a data in the United States, those soldiers who were involved in dismantling the nuclear warheads, although they had all the protective gears, even after that, they had high incidence of cancer than other soldiers. It means those protective gears were not enough. They might have that time, they were not enough. Biologicals, you might be knowing this Rau's sarcoma virus, very important story, and how it was discovered in the chicken. All of you might be knowing about this. So this is the one where you have biological cause of cancer. Actually, on this discovery, there have been several Nobel Prizes. So related to chemicals, pesticide is now also coming in the category of chemical carcinogens after metabolism. So just one example here, we were talking yesterday about green revolution. So sometimes we solve one problem and create another problem. The planning is not proper, it's not perfect. Usually what happens, that time the problem was related to the hunger. But later we found out that fertilizer and pesticides, the use was very heavy that time. You just see the area of Punjab is only 1.6%. If you look at the land, population 2.2%, and the pesticide use is 9.5%. This is current one. That time it was around 18% pesticide consumption only in one state. So heavy use of pesticide, now all of you might be knowing the effect. There was Malwa plateau that time, still there. But there is one train which runs from Bhatinda to Ganganagar, and it is called as cancer train. People have acronym for that because most of the people, they go for the treatment. There is a hospital over there for radiotherapy and chemo. So this is just a kind of planning where we see that we are solving one problem, but we are having another problem side by side. Then that time, use of urea, it was 3 times more than the national average. And in that area, you will find nitrogen poisoning and many problems related to fetal developments have been reported, along with several genetic problems as well as Like in some cases you will find more fingers or less fingers or miscarriage. Several defects have been reported in that. So even this heavy use of urea is not good. You have nitrate poisoning. So nitrate poisoning can damage a lot of genes and do a lot of things. Currently, Sikkim is the only state which has achieved 100% organic status. No chemical fertilizer, no pesticides. So this is a very simple foundation of cancer biology. All of you might be knowing. Initiation, promotion and progression. Initiation is the fixing of the mutation, proto-oncogene or tumor suppressor gene, to convert into the proliferating ones and then malignant. So look at this cartoon and tell me what you see in this cartoon. One is for cigarette pack, another one is mobile. So tobacco, all we know that it's not good. And tobacco is having a lot of chemicals which are causing. If you just look at the smoke of the cigarette, do you know how many chemicals are there? More than 3,000 chemicals are there in cigarette smoke, and out of that 100 chemicals are proven genotoxic and 50 chemicals people have used in the laboratory to generate tumors in the animals. So known 50 chemicals at least, they are there in the cigarette smoke or tobacco smoke, which we know that they can cause cancer. Now the second one is mobile. All of us are using mobile. Do you know that there may be some problem if we use it frequently? So there are some experiments now. People have shown that continuous, longer exposure of even mobile radiation, they are in less frequency. They are in different wavelength actually, so less energy. So even that can also cause DNA damage. So there are some papers. Just read and find it out. So what we should be doing, when you are not using mobile, then keep it away from your body. It is always recommended that it should be away from your skin, at least 2-3 centimeters away, so that the heating effect should not affect the body, other than radiation. So you never know, later you will come to know that some of the new health problems are coming because of this mobile radiation.
So, but WHO has classified the EMR of mobile in 2B category of chemicals, I think, and that says that we don't have the enough evidence to prove that the EMR of
that is what I said. We don't have enough, but we have some. Read those papers. There is one lab here in JNU, School of Environmental Sciences, and we do experiments in the rat, simple experiments. Keep the rat in the case, put your mobile on the top of the net, जाली होती है ना स्टील की, किसने सबने एनिमल यूज़ किया, तो उसके ऊपर रखा और उसको 2 घंटे तक ऑन रखा बस, and this went on just for few days, few weeks. Then all these things were observed. So this is reported that all those cells which are dividing, they are having extensive DNA damage, and they are exposed to the continuous mobile which is on. And again, sometimes you never know. Once you have proof, then we will realize that it is not as safe as it has been told. So again, we need to have a precaution. Only when you are using, just keep it yourself. Otherwise, just try to keep it away. Because when you have a mobile, when you are not using, it is communicating with the tower. So just one-way communication is happening. So the strength of the signal is, if it is 1x, when the phone is connected, you start talking, so now it is both. So signal strength, when you are talking, it becomes more than 2x. So that is already repeated. So when you are talking, then you are getting exposed to more radiation, more strength. Although it is in not that range where it is known that if it is X-rays or gamma rays, this is directly going to cause pain and damage. But longer exposure, even this frequency, it can cause, there are some people. So that caution should be taken. So this is the cartoon, that is what it is telling about. Maybe less, maybe there. This is again a very simple slide to tell you about concept of oxidative stress. RS, all of you might know. Left hand side you have scale, blue to red. Red is the higher one, blue is the lower one. Green is the balanced homeostasis. On right hand side you have cell cycle. And there are some causes also known how they can arrest the cells in different phases of the cells. So now, to have good health, we see that people are taking antioxidants and vitamins. So is it good to start taking vitamins or antioxidants? So unless you know that you need or you require, one should not take it. One small story related to this. Actually, I read that in the newspaper. In one area there was iron deficiency. It happens maybe endemic in some areas. So doctors were giving iron tablets to those people. Mostly they were ladies. So people were taking iron supplements for the longer time. After 1 year, doctors thought that let's also give them vitamin. Vitamin E is very strong antioxidant. And they thought that it will be better health. Iron supplement was already there. But when they took vitamin E, next day, most of them they fell sick. Now, this is a question for you to explain. As a scientist, just tell me what happened. Doctors thought that it will be good health if you combine two, but it happened otherwise. Why the fell sick?
Tissue production stop.
Maybe chelation.
Yeah.
Chelation.
Complex formation between oxygen and iron.
Complex formation between
Oxygen and iron.
oxygen and iron. Any other explanation? What could have happened? Doctors, they didn't know, they might not be knowing, but as a scientist you should know. Any guess, any idea what could have happened? Iron absorption inhibit हो गया होगा. Any other?
Imbalance between ROS and antioxidant.
Imbalance between
The antioxidant tissue and antioxidant.
क्या हुआ होगा ऐसे?
बैलेंस में अगर वो दोनों रहेंगे तो बॉडी सही रहेगी, पर अगर ROS ज़्यादा हो जाएगा तो ऑक्सीडेटिव स्ट्रेस होगा, एंटीऑक्सीडेंट भी ज़्यादा हो जाएगा तो ROS कम हो जाएंगे.
तो उसके रिस्पेक्ट में बताइए क्या हुआ होगा? now the question is how iron is stored in our body. Storage ferritin. Ferritin protein है store करता है iron. How iron is stored with ferritin? In oxidized form, Fe3 plus. So these people were taking iron supplements for a year or more than a year. So they had high store of iron in their body, the ferritin. When you give strong antioxidant vitamin E, what will happen? It will be reduced from ferric to ferrous. And that is how it is released from the ferritin. It comes into the circulation, taken by different enzymes or coenzymes, also the cofactor. So iron is circulated in the form of reduced ferrous. So what happened here? Iron stored was there very high. Suddenly gave high dose of antioxidant. There was a burst release of iron in the blood. So any metal, if it is more than required, it will cause metal toxicity. So what it tells? What is the message? The message is that never think that if two things are good, if you combine, you will get better result. Unless you know about their science, never go for combination. So when you are going to the doctor that I have a fever, they are giving you 2-3 tablets of the medicine, you know that how they work. So never venture once you don't know that how the combination is going. So that was the basis for what we are talking here. Tell me about this cartoon. What do you understand in this? One is the small cell, other one is the bigger one. The smaller one is stem cell, that is already differentiated. So what is the message in this? So this one is a little bit confused. And why it is confused? It is confused because of this. The purpose of showing this slide is not to remember anything. The complexity of the cell. One protein has multiple functions. You never know in which environment with which protein it is going to interact and what result it is going to give. You never know. So very small changes or fluctuations in the cell can give very different results. And when you do your experiments, you already know that how things are happening. Most of the time, you yourself get puzzled that I was expecting this, and I got something else. So in the cells, things are very complex. This is another level of complexity. Just to explain you, the circle is DNA and that strip is protein. You can take that as a transcription factor. We understand that promoter is there, transcription factor is binding, something is going to happen. But how that transcription factor is going to bind along the DNA, there are 360 orientation. In 360, there are a lot of permutations and combinations which are possible. So that is why when you do treatments to your cells, you never get 100% or full expression in most cases. Rarely that happens. And that can explain that how your protein is interacting with the DNA. It's A, B, C, D, 4 options are given here. And if this is the one that is very strong regulation, then slowly you will see weak regulation, weak expression. This is only 4, but there are 360 possibilities, more than that. So that is how in the cells things are working. Let's skip this one. Chemo prevention How many of you have heard this word? Chemo prevention. None of you? So chemo prevention is a word. It was coined in 1970s, I guess. And the idea was that how to control cancer, and that mechanism is going to be different from chemotherapy or radiotherapy. So chemo prevention means most of the things which is there being used in our daily routine, our dietary habits, how to use such items where you can modulate the occurrence or those events which are going to cause cancer. So mostly in this case you will find vitamins, some proteins, some fibers. So these are already known to prevent or slow down the growth of cancer. So here it is prevention, slowing down or reversal. These are the things which are associated with chemo prevention. Here are some plants which have been worked out. Now this is another important slide, since all of you are using animals in your research. Someone can ask question, why do you use animals? Just two simple answers. One is a similarity, where you will find even 90% of veterinary medicines, they are same what we take as a human. Only the dose is going to be different. If animals are there, the dose will be different. In terms of gene composition, 98% there will be similarity of genes what we have in the human. Plant for tumors. Plant and human gene is very different. Plant genome is very different from the human. So talking about that tumors. That is different. I am talking about animal models. That was there in the chemo prevention. Chemo prevention and chemotherapy. Even for the chemotherapy, most of the chemicals or the drugs, they were discovered from the plants, and almost more than 50%. The leads were there from the plant. Experiments were done over there, and then finally those drugs were developed. Here we are talking about animal models. What are the importance of animal models in biomedical research? So, comparable genes, these are the ones, and you will find the importance in the next slide, why it is important. This is just distribution. Mostly for the research, we are using mice, then rat, and there are some other animal species. But this is the importance of using animal models in cancer research or any biomedical research. So far, 116 Nobel Prizes have been given in the area of physiology and medicine, and the data suggests that out of 116, almost 100 discoveries could not have been made if those laboratories they didn't use the animal models. So 100 discoveries could not have been made. So that is the importance of animal models in biomedical research. These are some lists that you can check and find it out. Now another almost 10 minutes till 11 o'clock. So this is one very simple concept of tumor growth. You see blood vessels. What is the importance of blood vessels for tumor growth? So one hand to provide, on the other hand is how it was discovered. Blood vessel, just take the skin as a section. So you have basement membrane, and in the dermis you have blood vessel, epithelial layers and keratin layers. Tumor is growing. So this will grow only up to a certain size, and this will remain avascular unless it recruits blood vessels to become vascular tumor. So blood vessels are growing, and tumor is going to secrete some factors, then it is going to recruit new microvessels. So there was an earlier hypothesis that blood vessels are already there and they are getting dilated when the tumors are growing. But some of you might have heard about the name of the scientist, Judah Folkman. So Judah Folkman, he was in USA, in one of the laboratories, I think he was in Harvard. So he was doing some research on angiogenic factors. Actually, we expect that his cardiovascular research should be in the cardiovascular laboratory, but that was a cancer laboratory, cancer biology laboratory, where he discovered all these factors, vascular endothelial growth factors, then BFGF, his team only discovered. So they hypothesized that angiogenesis is essential for the growth of solid tumors. Unless the tumor growth is supported by the blood vessels, it will not grow beyond a certain size. And the reason, as you said, that it requires oxygen, nutrients and growth factors. So if you look at the stages of the cancer, normal cell to initiation, dysplasia, neoplasia, focal carcinoma, invasive carcinoma. So angiogenesis is required almost each and every stage. So if you target angiogenesis, you can target any stage of the tumor. So they hypothesize that having anti-angiogenic drug is better than any other drug, because you can target any stage of cancer. And there are more than 20 drugs in clinical trials. Some drugs are already being used to treat the cancer patients. So that is how you can also have angio prevention. So you can use those phytochemicals or those dietary plants where you know that they can inhibit the process of angiogenesis. Now if you look at small tumor, almost 2 millimeter the diameter, beyond that it requires the switch to be on for angiogenesis to grow further and become malignant. So till that size it will remain avascular. And there is a very dense network of blood capillaries. There is a limitation to which the growth factors and nutrients can diffuse in our tissue. And that is about 2 millimeter, not beyond that. So if any tissue which has to survive away from the blood capillary, it has to be within the range of 2 millimeter. Beyond that, enough growth factor and nutrients, they cannot diffuse. Neither the waste metabolic products and carbon dioxide, they have to be also removed from that cell. So both are required, both way traffic should be there, otherwise cells will die. And you can imagine the dense network in our body. Just take a needle, prick it anywhere, blood comes out. So it tells the dense capillary network in our body, how it is to support the survival of each and every cell in our body. So this is related to head and neck cancer, and you know that cancer stem cells are the main culprits. When the treatments are being done, some of them are surviving and the relapse is happening, and then you have growth of the tumor. So you might have heard about 2D versus 3D. These are some of the characteristics of the spheroids you might be knowing. But let's see how it works in the lab. I'll just tell you about one simple experiment that we have done. We took the tumor from one patient and then made single cell. From single cell we have grown the spheroids, and what we found that there are two phenotypes. In one case it is proliferating faster, in another case it is proliferating slower. During same duration, faster hyperproliferation and then hyperproliferation. So just remember these two words, hyperproliferative and hyperproliferative. Now, if you look at this data, see number of cells they were plated in each well and how these are growing. Hypo is the black one and hyper is the gray one. So in this patient having this single experiment, just growing spheroids from the tumor, what you can predict. We did one more experiment just to find out how do they respond to chemotherapy and radiotherapy. So here it tells that this black one is more, black one is hypoproliferative. So what will be the prognosis? We did some chemo and radio and we found out that the hypo one is more resistant to the hyperproliferative one. Those which are proliferating faster, they are getting killed easier by the chemo and radio compared to the less proliferating. So if any patient is showing high number of hypoproliferative, it means that person is not going to respond better to the chemo and radio. So that prediction can be made by just growing the spheroids from their tumor. And these are some other experiments just to know the cell types based on transcriptomic data. This is just to make you understand that how even the small tumor, that is a spheroid, just having 300 to 400 cells, how complex they are. Then you can imagine the bigger tumor, how complex they might be. What we did, we took single cell to grow the spheroid. So anticipation is that this should be clonal spheroids. If you do genomics, all the genes should be same, that was the expectation. But when we did transcriptomics, it was different. Now you can see epithelial, keratinizing, club cell, vessel cells. So all these 12 types of cells were there in one small spheroid, which was just having 300 to 400 cells. So what it is telling that when one cell is dividing, making a clone of a small spheroid, division of lever is happening from the very beginning itself. That is what transcriptomic pattern is telling in each and every cluster. So cells are very complex even in the small tumors. Now let me just tell you about one more concept. This is related to, this is the last one, mitochondria, all of you know. Mitochondrial exchange is now also one of the resistance mechanism in the therapy. So exchange, generally we think that it is going to happen for the energy. But here what has been found out that when you challenge the cells with the chemo or radio, those cells which are going to die they transfer their healthy mitochondria to the other cell, which is surviving. And that is how those cells, they become resistant, and even after chemo or radiotherapy, they remain there, they are not going to commit cancer. So that we have done several experiments to prove that it is happening in the cell culture, and similar things might happen even in the patient tumor treatment prevention as well. And long back, there was one paper published in the Cell almost 25 years back, and there it was proved that the cells which are dying by apoptosis or by any mechanism, usually they transfer the healthy organelles from them before dying to the other surviving cells. So again here there is a concept of survival of the clones or the colonies where they are. This is what we discussed again. That if a group is being threatened, then some of them are going to sacrifice themselves to make others survive, so that the progeny will remain there. So that kind of situation is always there in case of cancer cells. Is there any question?
Mitochondria fully transferred to person. Mitochondria fully transferred to person.
Mitochondria will be transferred fully, means there will be fusion in the mitochondria and some portion is going to be transferred. But number of mitochondrial transfer is there. Full is also getting transferred. So there are different cargoes which are being transferred from one cell to another cell. Mitochondria is also one. Actually, this was one, I don't know, but video will not work here. But this is the live imaging. This is the leavened mitochondria, red one, and this is the connecting tube, nanotube. You will find from one cell to other cell how this is moving. But I don't think this will not work. This is a video to just show you how the mitochondria is moving from one cell to other cell through nanotube. So this can be worked out very clearly. Any other question? The healthy one is going to survive. This is for the survival. You will see that oxphos is going to produce more ATP, which is needed for the survival. The other one may die, which is transferred. So usually this phenomena has been survival. There may be other biological events as well. But in this case, when you challenge them with the stress like chemo or radio, it will be for survival. Any other question? Thank you.
What are the techniques and the methods we are trying to maximally, I am not going to say eradicate the cancer, but maximally create the cancer? What are the ways we are?
Maximal eradication, you need to see the targets that you ask, and accordingly a combination of the drugs, so that you can target maximum cell population. Because very important concept is tumor heterogeneity. So in case of brain cancer, earlier it used to be single biopsy. You will take one biopsy and then on the slide H&E staining, find out the stage and accordingly they did the treatment. It was not working. The reason was that in a whole tumor, if even one clone is amplified, there may be, suppose if there are 10 clones, there are 10 different tumors in a given tumor. If you take biopsy from one side, you may get samples from 2 or 3 sometimes. Others will be left out. So your treatment is based on only this. So 70% is left out. You never know what is there in the tube. So nowadays it is multiple biopsy. They put the needle from different sides to map the tumor and see what kind of cells are there. Accordingly, they try to have the drug combination so that multiple cells, multiple cell types can be targeted to get the better therapeutic response. So this is current stage of treatment.
So then also mRNA vaccines.
Yes, mRNA vaccines, DNA vaccines. So vaccines are of usually 2 types, all of you might be knowing. One is called as preventive vaccine, another one is therapeutic vaccine. So in this case it is therapeutic vaccine, where you are trying to target cancer cells, having some target so that they can go there and kill those cancer cells. There are some vaccines which have been developed. But again, because of this diversity what we have discussed, the efficacy or the treatment response what we are anticipating, it is not coming. It's always partial response. That is what we are doing. Anything else?
सर एंडोक्राइन ग्लैंड के कैंसर में हार्मोनल थेरेपी बेटर है या सर्जरी? मतलब
सर्जरी तो हमेशा बेटर है, दिस सर्जरी से रिमूव हो सकता है completely.
हार्मोनल थेरेपी से
Hormonal therapy again, for example, mostly it comes for the prostate cancer, ovarian cancer, breast cancer or some other endocrine glands. So hormonal therapy is going to work only when there is an imbalance. And again, I am not sure that it is going to remove those genes because of which that cancer is there. So hormone could be just for the maintenance. It can categorize under the maintenance therapy. The way we have blood transfusion. You have blood cancer. You might have heard that patient is going for blood transfusion every 2 weeks or 3 weeks. So it is just maintenance therapy. This is not true. Because those cancer cells are sitting in the bone marrow somewhere and they will keep producing those blood cancer cells. So that will be there always. So still it is not going to cure. It may maintain up to some level this growth and formation. Thank you.